Hello and welcome to the Live from the Lab series. Today we'll learn about how early nervous systems might have evolved, why we have many different types of nerve cells, and how complex animals' brains came to be. My name is Siri, and I'll be the moderator for tonight's episode. And this event is the fourth episode of a five-part series, which is guided by the overarching question: what did animal ancestors look like? The speakers in this series, as well as myself, are all connected through the Evercell Marie Curie network and a common interest in the genome, cells, and animal evolution. And so let's get into tonight's topic, the evolution of the nervous system. Our nervous system, that includes the brain, controls so much more than just what we think and feel, how we remember, talk, or move. But moreover, it controls things that we are less aware of, such as our heartbeat and the digestion of our food. But how did such an incredible and complex system of cells evolve? The speakers of tonight are not researching the human nervous system specifically, but will be able to give you an insight into their research on the nervous system of a marine worm, sea urchins, as well as the lampreyfish and the mouse. Tonight's speakers are Milena, Periklis, and Francisca. They will take us from less to more complex nervous systems in animals. With that, I will pass you over now to Milena, who has the honor to begin the talks. And if you've watched the previous episode, you'll probably recognize her as she organized this event with me and also co-moderated the series with me so far. But tonight she is the speaker. Milena.
Welcome everyone. I am Milena and I am here in a basement at the University of Exeter because later in the talk I want to show you my microinjection rig here behind me. And I do tend to talk fast when I'm nervous, so to the people from the future who are watching this on YouTube, you can go bottom right corner and put me on 0.75x speed. And to the rest of us in the present, I'll do my best and good luck. I am going to talk to you today about movement and how movement evolved. And we mostly associate movement with animals, but it's not actually unique to them. And like all life and all animal life, movement started in the ocean. So if we want to find out how it evolved, we need to look under the water surface. And in the sea, there are some animals that don't move very much, like these corals. Then we have animals like clams that look deceivingly immotile, but they do actually have muscles and they can use them to jump around the seabed. And then we have jellyfish and fish that swim, but using very different modalities of swimming. But movement didn't start out looking like any of these, and it isn't limited to animals. So even single cell organisms like bacteria can swim. And this single cell creature is not a bacterium, it's called a cranoflagellate, where crano means collar, which is this yellow stuff here that it uses to catch prey. And flagellate is for flagellum or cilium, which is this whip-like thing here that it uses as an oar for swimming. And you'll see that this kind of swimming is actually very popular in the sea, in both single-cell animals, in single-cell organisms, and in animals as well. And it's important to note that these cranoflagellates are famous among scientists because it is thought that all multicellular organisms came from an ancestor that looked somewhat like this. So basically, we think that we had one cranoflagellate like this that then organized into a colony of multiple cells like this. And then eventually the colony members will have lost independence and decided on a division of labor and became the first multicellular organism. So animals originated from ancestors that moved in this way, and we can find it to be a very widespread type of movement among animals today. And you might not have encountered animals that move with cilia because most of the animals you can see with the naked eye move with muscles, and cilia are reserved mostly for movement of small marine animals. So what moves with cilia. I told you already that single-cell organisms like the chronoflagellates from before, they swim using one or more cilia that move in waves like this. And sponge larvae also move entirely using cilia until they grow up and become sessile. But in fact, most animals in the plankton can do both. So when they're larvae, here pictured there are many different larvae, they swim in the plankton using cilia, and then they fall to the seabed and they become oysters and sea urchins, for example. Or in this case, this is the animal that I work with. They can grow up to become this here worm called Platinerace, and this is the larval stage that I work with. And this belt of hair here is the ciliary band that it uses for swimming. So because it's so widespread in all these distantly related animals like worms and sea urchins and oysters, we might assess that moving with cilia is a very old behavior. And another important thing about these cilia in animals is that there are many of them and they all beat together in waves, which is something that has to be orchestrated through the nervous system. So we have a very old behavior that must be regulated by very old networks in the brain. And I work with these animals because I like thinking about evolution and about how life went from having no brains at all to where we as humans are today. And the first step in that process was actually inventing nerve cells and inventing the nervous system. And there are many different theories about how and why that happened, but one of them postulates that the first neurons appeared to control ciliary swimming. And again, we know that cilia are quite old and quite widespread, and that so must be the neural networks that control them. So let's now zoom in on them. Here is Platinerace with its ciliary band and see what this theory says. So this is how it might have happened. So at first, cells with cilia that were responsible for movement would get chemical signals from neighboring cells that would sense whether a predator is nearby or whether food was nearby, for example. And this way, these cells would know whether to move or to stop moving. But this kind of signaling is not very precise and not very efficient. So it works for simple animals like sponge larvae, but throughout evolution, animals got bigger and they had more ciliated cells that needed to be coordinated and that needed to get more precise signals. So then the sensory cells, instead of just diffusing signal molecules around, started developing extensions like this in order to contact other cells in a more direct and in a more precise way. And when they developed extensions that we now know as axons, they could start contacting more than one cell, giving them the same signal so that they can all do the same thing at the same time. And this is how the first sensory motor neuron would have appeared, having both a sensing function through a cilium and controlling the behavioral output by connecting with the other ciliated cells. And in marine larvae, sensory motor neurons are quite common, but later as animals got bigger and bigger and got more and more neurons, more division of labor occurred and neural networks became more complex and a subdivision into motor and sensory neurons would have appeared. Okay, so now we are live from the lab, right? And I said I will show you my microinjection setup that I use for CRISPR microinjection. So CRISPR, as you may have heard, is this relatively new gene editing technology, and the scientists who invented it just got a Nobel Prize this year, so it's quite important. And it works basically like targeted molecular scissors. So if you put CRISPR molecules around your target DNA and instruct it cut here, it will cut the DNA there and the gene it cut will stop working. And this has also lately been adapted to insert new pieces of DNA in the targeted spot. So here now is how I do it. Hopefully you'll be able to see everything. Here I have a little dish that's full of fertilized Platinerace eggs that are on a stage made of an agarose gel in a ridge so that they're relatively immobilized. And then here I have a very thin glass needle that I fill up with the CRISPR mixture, and that then I put into the dish and find the eggs with it. And then I have this joystick-like thing here, which I use to move the needle around and go from egg to egg. And it makes it like a sort of repetitive little video game. And on the other side here, I have a pressure unit that is used as a sort of plunger for the injections, and it's controlled by this here mouse. So when I press the mouse, the CRISPR mixture will enter the eggs. And then the CRISPR cuts the worm's DNA, and when they grow up, one of their genes won't be working. And to go back to my presentation now. So yeah, this is how it looks like under the microscope. So I could have used the stock video here that's better recorded, but this one's actually mine. And what you can see is what not to do. This is what happens when you inject too much liquid into the worm egg, and it explodes. This is also a good way to check if the needle is clogged, because obviously if it is, the explosion won't happen. And to go back to evolution now, what do I use CRISPR for? So I want to turn off a gene that makes the worms dopamine. So you know that dopamine is the reward chemical in humans. When it gets secreted when you eat chocolate or when you do cocaine, but in the worms, we think it does something else, that it actually gives the cilia a sort of heartbeat. So much like the human heart, the worm cilia have an automatic rhythm. And because this kind of pacemaker circuit is very widespread in marine animals and very old, if and when I figure out how exactly it works, it might give us some hint about how the nervous systems of ancient animal ancestors worked. And that's it from me. So we organize these talks so that we go from the farthest relatives of humans to more closer relatives of humans. Mine was the farthest relative, and our next speaker is going to be Periklis Paganos, who works with sea urchins that are a closer human relative. Periklis, if you want to start.
Thanks, Milena. Hi, everyone. I'm Periklis Paganos. I come from Greece, and today I'm live from the historic marine station Stazione Zoologica in Naples, where I do my PhD. Talking about my PhD, let me introduce to you the animal I'm working on, which is this very beautiful Californian sea urchin species. But actually, I'm not working on this life stage, which is the adult stage. Instead, I'm working on early life stages like the ones you can see on the screen behind me. So these life stages that look like a weird Eiffel Tower are called larvae. And actually, these larvae are very tiny. So that's why I'm using a stereo microscope to show them to you. And now that you're going to know the real star of this talk, let's go to the talk. So let me share my screen. Okay. So when talking to non-scientists like my best friends about my PhD project and what I do for a living, they always ask me the same thing. Periklis, do sea urchins have neurons? And if so, do all animals have neurons? And the short answer is yes. Animals have nervous systems that differ in complexity, but nonetheless, they perform similar functions. Such functions are, for example, the control of movement, as you've heard from Milena's talk. Neurons can sense environmental changes. They can be involved in the growth of the organism, but also control different processes. Despite the differences of the nervous systems in complexity, all of them have one major role in common, which is to transfer information between cell types. And now let's take a step back and try to understand how different animals are formed. On this slide, you can see two morphologically identical eggs that even I wouldn't be able to tell them apart if it wasn't for the labels. And mysteriously, somehow these two eggs will give rise to two completely different organisms. But actually, this is not such a mystery. Their genetic material, their DNA, is responsible for this. Here, you can see the same egg, and inside the egg in purple, the DNA inside the nucleus. DNA acts as an instruction manual that provides the cells with all the information to form an organism. This information is encoded in smaller pieces of DNA called genes. Different organisms have different operating manuals, but nonetheless, all of them are written in the same language, in nucleobases. And let's see an example of this. Starting from a fertilized sea urchin egg that you can see here, the single cell will proliferate and diversify to give rise to different complex structures and life stages. This is the larva that I showed you before, that will eventually lead to the formation of the sea urchin we all know, in a process similar to what Ines described last week. But now also within the organism, cells commit to give rise to diverse cell types. This is because different cells can only read parts of this manual. And here are some examples of distinct cell types present in the sea urchin larva I'm working on. So for example, here you can see cells producing skeleton that is used for support, ciliated cells used for movement, muscle cells that are responsible for contraction, and gut cell types involved in food processing and digestion. And so far, I've already mentioned that all animals have neurons. So surprise, surprise, sea urchin embryos and larvae have neurons as well, that you can see now depicted in green. What is very easy to see is that at the beginning of development, there are only a few neurons present. As you can see, there are only a few green spots. And as development goes on and the animal becomes more complex, the whole animal looks to be green. This means that as the organism becomes more complex, needs more neurons to coordinate different functions. Another fascinating thing is that nervous systems, apart from being able to perform similar tasks, they're also able to produce similar chemicals, which is also probably partially the reason why they can control similar functions. So on this slide, you can see molecular biology stainings that either result in fluorescent or color-based signal to detect such chemicals. So for example, these two chemicals, serotonin and TRH, which are involved in different biological processes, are found to be present in a variety of animals, including us humans. That being said, what I'm trying to do in my PhD project is to understand which part of information the sea urchin neurons can read and compare them to neurons from other animals. This will allow me to eventually understand how similar neuronal types arose during evolution. To do so, I isolate single cells from the larva, which now you can see in gray, and extract the information from them. Then this information is computationally analyzed, and this results in an artificial reconstruction of single cells, which now you can see as colorful dots. Single cells that have the same information are grouped together into clusters to represent cell types. And this is a very powerful method because it allows me to recognize the cells of interest. So for example, here, you can see in blue the cells that produce this chemical I talked to you about before. And by doing this, first of all, I can easily recognize them from the pool of cells, extract all the information that is within them, and this will eventually help me understand more about their identity and evolution. That was all from me. I hope you enjoyed, and I really hope that now it's clear that us animals share more features that you might have thought. And now let's continue this episode with Francisca, who will talk to us about the evolution of the brain. Thank you.
Hi. So I'm talking to you to our lab here in Heidelberg University in Germany. And now I would like to share my screen with you. Okay, there. Okay. So hi, good evening. In this session, I will talk about the evolution of the brain in vertebrates. But first, I would like to introduce myself. I'm Francisca Hervas and I live in Germany, but originally I come from Ecuador. where I live most of my life just enjoying the beauties that a country like this offers to a nature lover like me. But because I don't only love nature, but the science behind its evolution, 2 years ago, I crossed the Atlantic to start my PhD in the University of Heidelberg, and basically got involved in research about organ evolution. So currently, I'm studying one of the oldest questions in natural sciences. Where does the brain come from? Now, I don't study the human brain, and before you ask, I will explain why. It's not that I don't find the human brain interesting. On the contrary, our brain is remarkable. However, this does not mean that the human brain is the pinnacle of evolution. This is a misconception led by the incorrect belief that humans themselves are the peak of evolution. But in reality, nothing is the pinnacle of evolution. Let me explain with this graphic. Here, I show you a tree that illustrates the evolutionary relationships of groups of vertebrates. And at one side, there's a time scale. So the species aligned with time 0 at current living species, the ones we see today in the world. These species are linked by their ancestors. Ancestors that live somewhere back in time, here represented by these orange circles. So if you take a look at when humans appear, you can see that we are not such an old species, which means that we haven't evolved independently as long as other older species. And this implies two things. First, some non-human species have structures that we don't. There's nothing in the human brain, for example, that has the capacity of processing UV vision or skin camouflage. And second, this means that the human brain has retained ancestral traits. Even in the scenario of having innovation, much of our brain components evolved before the appearance of humans as a species. So focusing only in humans as an isolated species will lead to miss most of the evolutionary history of the brain. Now, my project specifically aims to understand how the brain of the ancestor of all vertebrates look like. The one that is represented here by this circle. How are we doing that? Well, in the absence of a fossilized brain from this ancestor, we have to use information of current species to infer ancestral traits. To do that, we have carefully chosen the species to study. And for the first comparisons, we picked the lamprey and the mouse. Why? Well, let's plot these species in the same tree that we saw before. As our purpose is to infer this node, we should pick a species from these groups coming from these two branches. This group here is the first one that is split from other vertebrates, and the lamprey is in this group. The other branch led to more diversity. So in order to have a good picture of the whole vertebrate history, we pick one animal from the last group that split, mammals. Now, between these two species, the groups, there's a lot of evolutionary distance. So from all mammals, we pick the mouse because for this animal, there's a lot of information available, and this alleviates a little the challenge of comparing distant species. So how exactly we do compare between species? Well, we must be sure that we are studying the same thing in both animals. And this is not so trivial because the brain is a multi-part organ. If we want to be sure that we're comparing the same structure in both species, we have to go for the constituents of these structures, cells. Cells are the bricks that organize themselves in tissues leading to organs. And specifically, we use genes as the units of comparison. We identify the genes that are active in each cell at a specific moment. The activation of a set of genes, plus where, when, and how much they are activated determines the identity of a cell. And changes in these activation patterns can be associated with evolutionary change. So our task is to identify differences in these patterns of gene activation between species. Now, how do we get this information? Well, we take the animal and dissect the brain. Then we dissociate the brain in order to separate cells, kind of like making a soup of cells. And then this soup is loaded in a machine that labels each gene being expressed in each cell. So with this information, we do the following steps. First, we determine the cell identities by identifying which cells are present in each animal brain. Then we estimate how related cells are between species. Basically, we look for similitudes and differences. And at the end, with this information, we infer the most possible cell composition of the ancestral vertebrates. So, what have we found so far? First, we must notice that cells as other biological entities evolved. Therefore, individual cells cluster in cell types. And cell types cluster in bigger groups of related cells, kind of like cell families. So we found that the cell composition of the brain in the two species involves the same cell families, but the mouse has more numerous families because it has more cell types than the lamprey. Because of this, we think that the ancestor of all vertebrates had already this cell composition, these cell families. However, through time, these cell families have evolved, leading to the increase of cell diversity that we see today, at least in mammals. Now, we're getting closer to understand how the brain of the vertebrate ancestor looked like. But this is just the beginning and the setting of the baseline. In the future, by studying more species, we can assess how exactly this cell diversity changed through time. We can explore how changes in the cell composition of the brain accompany vertebrate evolution. And I really hope that in the future we can meet again and discuss further about this. But for the time being, I can only thank you for your attention and of course be open to any question.
Thank you for this really interesting talks. Like all of you had very beautiful presentations and I'm very impressed. And so now you can all ask questions. You can do that inside the Zoom chat and inside the YouTube chat. And for now, I'll start off with one question. And I have one question for Milena. So now I'm allowed to ask Milena questions and she's the one who has to answer them. And so you showed some CRISPR technology, and I was wondering how difficult is it to use, as it's been a lot in the media, and also what are the ethical issues with it?
Yeah, so actually the biggest problem with CRISPR is exactly that it is so easy to use, and as opposed to previous gene editing technologies, it's much more amenable to being scaled up. And on one hand, the fact that it's so easy is a big advantage, especially for people like me who work with unconventional model organisms, because earlier methods of generating mutants were quite hard to pull off, especially when you have an animal that, for example, doesn't have a very well characterized genome. And now that's easier and it's also quite cheap. So scientifically, I think it's a really good thing because it allows genetic manipulation in a wider range of organisms, and it also contributes to the democratization of science, because now researchers in developing countries with more limited science funding have access to a really good tool. So that's not where the ethical issues are. Like my mutant worms will never get out of the lab and mess up the ecosystem. We take precautions against that. And also knowing their nervous system pretty well, I can be pretty sure that they don't suffer when I make them into little mutant monsters. And yeah, in other words, I am not the problem here. The problem with generating such an efficient and affordable tool is that as it becomes democratized, it becomes subject to capitalism and the growth mindset and our ability to turn a blind eye to suffering. So we have shown already as a society for many decades that we have absolutely no problem using selective breeding and medication to engineer better farm animals and dogs even, that often suffer immensely because of how we modify their bodies. And then what will stop us from using increasingly advanced methodology in order to generate organisms that will serve us better? I don't really think there is such a thing. And moving from farm animals to people, as long as there is money to be made there, and as long as the people who prioritize profit remain the only ones with power and with a voice, I don't think we're safe from a future in which the human body and the human embryo will be subject to modification, which will first be under the guise of health improvements, and then who knows what, like creating superhumans or some other futuristic form of eugenics. All in order for someone to completely unscrupulously maximize their profits. So those are the ethical issues, in my opinion.
Thank you for your thoughts on this. They were very thorough and interesting. And I will continue with another question. And this one is for Periklis. I was wondering, why is the sea urchin such a famous model organism? So the animal you're working on has been researched on for many years, and there's been already so many findings. And I was wondering why exactly the sea urchin, and yeah.
Thank you, Siri, for this question. Yes, indeed, sea urchin is a celebrity in developmental biology. Actually, it was one of the first animals to be used to study development. And this is actually because, first of all, the embryos are transparent. So this means that all the cellular movements and processes can be easily observed. And some of the landmarks in terms of in vitro fertilization have been described in sea urchin. For example, the fact that one egg, one oocyte, can get fertilized by one sperm, it was first described in sea urchin. The fact that there are maternal determinants in the egg that will help the organism to grow was described in sea urchin. The fact that chromosomes in the nucleus regulate cell fate. And actually, this is very interesting because many of these findings have been done here in Stazione Zoologica almost a century ago, more than a century ago. So it's very nice to ask me this question.
Thank you. Yeah, we work at a very famous Stazione, and a very beautiful place in Naples. I can highly recommend everyone to go there, and if you're ever in Naples. And so I have my next question is to Francisca. And so you were saying that the mouse has a higher number of cells. And I was wondering why the lamprey fish has not the same amount of cell types as the mouse, because the lamprey fish had more time to evolve, you could say, or at least independently. Yeah, what is your thoughts on that?
Yeah, thanks, Siri. So this is a really interesting question, because I think this is like the next step. Now we know that, yeah, the lamprey has less cells, the mouse has more cells. Now, yeah, of course, it would be amazing to know when and why it happened. But of course, this is a difficult question to reply just like with one answer, because we don't know. We can only like infer from what we know. So yeah, the mouse has more cells, the lamprey has less. That means that whatever happened in the lineage that lead to mouse or to mammals didn't happen in lamprey, or the lamprey couldn't evolve more cells. So it is tempting to believe that because we're talking about the brain, in mammals has this, I don't know if complex is the word, but you can call it this particular or conspicuous behaviors, and also a social structure. It is like tempting to believe that maybe it's associated with this, but we don't know. So this is just like where you might think, but it is clear that the lamprey has survived all this time without more cells. So there's no need maybe for these animals to evolve more cells, or there's a restriction. In any case, for the mouse or for mammals in general, it has been quite successful to evolve a higher diversity of cells.
Thank you. And there's another question for you, Francisca. And so someone is asking that in your presentation, there are signs on the slide where you showed the sea lamprey fish. So probably the arrows indicating common ancestors. Do you know what these common ancestors, these creatures are, and yeah, how they look like?
So no, I mean, from some ancestors, in animal ancestors, there are fossils, so we can like more easily infer the morphology of the form. When you talk about cells, of course, even when there is fossils, these soft tissues normally don't fossilize. So it's unlikely to find a brain, for example, fossilized. So what we're trying to do with this studies is like you're trying to infer basically how the ancestor most likely look like based on what you know from a species that live. So if you have a good representation of a species that you study, in our case, the vertebrates, you can say most likely the ancestor of all vertebrates had this. And if you compare with animals that are not vertebrates. So this is actually the goal, like to infer this, but there is not fossilized brain of the ancestral vertebrate. So we cannot, of course, be super sure how the brain look like. We can just infer based on what we know from the species we're studying.
Thank you. And I will go on with the next question for now to Milena. And what other scientific techniques are you using? And is there anything else you are injecting?
You can actually inject loads of things, and it's all very useful. So I'd say the most common and the most effective, reliable things you can inject are different constructs that shut down a gene's function. So that would be CRISPR, like I said in my talk. But you also have something called RNA interference, where you inject small RNA molecules that bind the target RNA and disable its ability to make proteins. And a similar thing are morpholinos, except they are synthetic molecules and not RNAs. And then another thing that you can do, but it's a bit more fiddly, is inject a construct with a tag that will make cells fluorescent if your target gene is active in them. And more cool things. One is called photoconversion. So you inject molecules that make the whole organism green fluorescent, and then you shine a light on a specific part of the organism, and only that part becomes red fluorescent. And if you did this in an animal embryo, you can, for example, track its development and find out which structures will come from only that specific part that you photoconverted. And last and most important for neurobiologists is GCAMP, which is a construct used for calcium imaging. So this is when you inject, again, a fluorescent construct, and as it becomes active in the animal, it will give you fluorescent signals in when calcium gets released in neurotransmission in synapses. So this is really super useful if you want to know which neuron is active and where and when and in response to which stimulus. So I'm sure there's plenty other methods, but these are the main ones that I've used throughout my research.
Thank you, Milena. And there's another question in Zoom from Sarah, always having great questions, and I'll just read it for you. So a wild question, but assuming that it would be possible to swap the brains of animals. So for example, the two Francisca discussed, so the lamprey, fish, and the mouse, and the nervous cells from the same cell families would take care of the same functions in the new organism. Would the animals still be able to survive? I mean, I wouldn't think so in the case of the mouse and the worm thingy, right? Even though they share some similarities. But how closely related do two organisms' brain have to be in order to do this to be possible, hypothetically speaking? Very big question, very interesting. Who wants to have a go?
Yeah, I mean, this is really interesting question, because I mean, this is something similar as what actually functional biologists do to try to understand how evolution works. So first of all, it is not so it's not easy to swap the cells from one animal to another, but you can try to mimic what happens in one animal in the other animal. So for example, if the mouse has more cells and we manage to identify what's the genetic and the embryological mechanisms that lead to these cells, we can try to use techniques like the one Milena was talking about, CRISPR-Cas, in order to make a lamprey that looks more like a mouse. So to see like how possible it is. So if this, to try to understand if this happened in evolution. And this is not about the closeness of a species. Of course, in closer species, sometimes it's easier, but there's like experiments of like that when they have, I don't know, like compares, for example, snakes and mouse, or the Darwin finches and with normal chicks. So these kind of experiments are being done in the daily basis, I will say. So it is possible. And actually that will be one of our ultimate goals in the future, if we identify what truly makes the brain of the mouse, have more cells, we can try it to make a lamprey that mimics the brain of the mouse. I don't know if this animal will be truly able until when to survive. Sometimes you just have to sacrifice the animals before they become adults for reasons of ethics. But if not, yeah, this is something that we will see in the future. But it's not a crazy question, by the way. This is actually something really cool to study.
I'm blown away. I haven't thought about that before, and also thank you so much for the answer. Yeah, I'll continue with another question. Periklis, a question for you. And have you made any interesting findings about the neuronal activity in sea urchins? So you're already nearly finished with your PhD, right? So you might have the most findings, maybe?
Well, yes, we have something. So first of all, we were able to recognize all the cell types in the larva, which is already a huge thing. And neurons were among them. We found a great number of neurons present at this stage, which has never been described before, at least this high number of neurons. And in general, in our team, we have some evidence that at least 2 of them are involved in the growth of the larva, which is very interesting. And actually, this is very nice because this gives me the opportunity to say something else. I said my team because I want to really highlight that research is a team effort. People usually think that as scientists, we always compete and we hate each other, but in reality, we learn from each other, we collaborate with each other, and this is how we do it. And this is something I think people have to know. I deviated somehow from your question, but I kind of replied.
No, that's completely fine. And yeah, I completely agree. I visited Periklis lab actually, wow, 1.5 years ago. And I was truly amazed by how you all work together and discuss and everyone is just super fascinated and has so much enthusiasm. And yeah, I really loved the way you were working together inside of your lab. So thank you for the answer. And I will continue with another one for Milena. And I was wondering if the environment has an influence on the ciliary beating, difficult word.
Yeah, it is an uncommon word. Yeah, loads. So all animals respond to their environment, and in the case of these marine larvae, they don't actually have that broad of a repertoire beyond responding with movement. So starting with sponge larvae that don't even have a nervous system at all, they still send signals directly from sensory cells to the motor cilia, so they can move, for example, responding to light. And this response to light is very common in going from, bacteria to unicellular organisms, and then in a different way in plants as well. So they all react to light, and that is called phototaxis in animals, or phototropism in plants, where they grow towards the light source. And animals don't necessarily move to the light source. So it depends on the species and the developmental stage. Some of them can move to the light, some of them can move away from the light. And these signals that tell them go to the light or move away from the light, they can be transmitted either directly from sensory cells to the cilia, or indirectly through some sort of visual neural circuit. And apart from light, it's also common that they react to chemical signals. So, for example, if they sense that oxygen is low, they can adapt their swimming so that they try to move somewhere where there's more oxygen. And very importantly, they get chemical signals from the environment that will tell them now is a good time to settle on the seafloor and to metamorphose. And this will also change the ciliary swimming so they fall down. Yeah, but to go back to my pacemaker, like heartbeat circuit that I work with, that one's actually active all the time, or should be active all the time, even in absence of environmental input. And the environment only modulates it rather than activating and deactivating.
Cool, thank you. And I have another question for Francisca. And so you work with the lamprey fish, and it looks so scary. It has so many teeth, and I was wondering if you're scared by it, and how is it like to work with the lamprey fish?
So it is an experience, no, or at least at the beginning it was an experience, because two things. First, of course, professionally, not always like anyone, I think no one from this network works with the species that are like easily to breed, or as easily to breed in captivity, or as other species that are normally used in biology. So for this animal, it has well, not a complicated, but a life cycle that involves many stages. So that means that we only have one shot every year to sample the embryos and the adults. And this leads to, and you depend a lot of environmental forces, because when it rains too much, maybe there's no animals, when it rains too little, and so on. But this also leads to collaborate with people in other countries, in other labs, and this is professionally and personally really nice. And also to plan ahead a lot the things that you have to do. And personally, I think for me, the perception of what is not nice has changed by working with the lamprey. I have been aware that how the perception of what makes you feel uncomfortable changes, because of the rows of the teeth at the beginning, I was feeling a little bit weird when I was looking at this animal before I even started my PhD. And now I have even take the animal from the mouth, and I don't really mind. And I think this says a lot about how your brain change perception about what are the things that make you feel uncomfortable. You just have to, deal with it and get used to it, I think. So that was nice.
Thank you. And I have one final question for tonight, and this is to Periklis. And I was wondering, so your sea urchins you use are Californian sea urchins. And I was wondering if there's a reason behind using this kind of specific sea urchin. So it's a bit maybe alike than why is the sea urchin so famous, but yeah.
Isn't the fact that they're gorgeous enough? Actually, no, there are many reasons. This sea urchin species was the first one to be sequenced. So all the genomic tools are there. So actually it's very convenient to use this one. And there has been a lot of work describing how it develops, how different cell types are formed. So it's very, very easy to start working on it and start building brick by brick a story that has been ongoing for many years. So it's mostly convenience. For me, it's because it's beautiful.
Thank you. And yeah, I imagine like a sea urchin, like just a very big book where you can look up a lot of things and yeah, and hopefully also relate it to maybe other animals and learn from it. There's another question, and Laura asks, who else thinks that Platinaris is uglier than lamprey?
Nobody thinks that. Nobody thinks that. I know it.
I do.
Okay, if there are a lot of opinions inside the chat, nobody thinks that, nobody. And okay, well. Ooh, there's some questions, I'm very sorry, on YouTube. Do you have a bit more time? I will ask them and then we'll go for tonight. So Danielle asks on YouTube, will it be possible to follow all the steps that have been followed throughout the evolution of the nervous system? For example, the cerebellum, if jawed animals have it and lampreys, so jawless, don't. How can we investigate the origin of the cerebellum? Can we understand how it originated without having access to the ancestor's brain, only inferring from currently living animals? That's a really good and interesting question.
That's for me, I don't know, because I sent some. Yeah, so thanks, Danielle. This goes directly into one of our research interests, actually. And yeah, I mean, I guess you can never be super sure that the ancestor of these animals had a cerebellum or not, because we don't have access to the actual ancestor, but we can be like as positively sure as possible. So far, there's no much evidence that the ancestor of vertebrates had cerebellum. And we don't find genetic evidence of that neither. But that does not mean, of course, that the ancestor didn't have the cerebellum. It's just like we can assume, well, not can assume, we can just like suggest that most likely the cerebellum appear later. Because of the functions of the cerebellum and because of the fossil record that does suggest a little bit that the cerebellum evolved later, we can infer that it happened later. However, like, I don't think we can be 100% sure of how the ancestral vertebrates look like, of when exactly this did happen. We can just be as sure as possible. And I think that's one of the reasons why every 10 years you have like new discoveries, because new methods actually are developed, and these allow to go to understand better evolution. But maybe I'm wrong, but so far I don't think there's like 100% evidence or methodology that allows us to know 100% if the ancestor had it or when exactly appear. But yeah, all the evidence points that out that appears later, the cerebellum.
Thank you. And with that, I think we can leave it. It was very nice, and also thank you to all the speakers for giving such really fascinating insights into your research. And yeah, Milena looks very skeptical, not so sure about it.
No, I'm super excited. I'm moderating tomorrow, and so yeah, please come, and thank you, everyone.
Thank you, and see you tomorrow, hopefully. Bye.
Bye.